Hello, my name is Eric Lee. I'm from BC Cancer Agency and the University of British Columbia. And today I'll be giving a talk on my paper, ESQmodel, Biologically Informed Evaluation of 2D Cell Segmentation Quality in Multiplex Tissue Images. Alright, so to start off with a brief background, I would like to briefly talk about antibody-based proteomics. So classical methods to explore the heterogeneity in tissue ecosystems have leveraged flow or droplet-based technologies to define cell subsets and cellular crosstalk. A key problem here is that this requires tissue association, as we can see over here, which really disposes the spatial context that is required to understand the underlying cellular interactions in structural organizations. So in order to probe in situ biology with sufficient depth, we can now use antibody-based multiplex protein imaging methods such as imaging mass cytometry and multiplex ion beam imaging, which is MIBI. These don't disrupt the tissue integrity and really preserves the spatial context when measuring expression intensity. So this allows us to localize proteins to study their distribution and really reveal the interactions between cellular types with applications in stratifying disease subtypes and finding potential therapeutic targets. So to demonstrate the importance of spatial context, we can compare a UMAP visualization and a spatial neighborhood graph that are labeled by their colors. And these colors represent different cell type clusters. So we can see here that there are non-random spatial patterns of cell-cell interaction in the graph. And then if we study the spatial signal, this really sheds light to an extra layer of spatial signal on top of current studies of mainly just cellular neighborhood analyses. So with the spatial data required, the focus of the spatial experiments lies in performing computational analysis. So the analysis workflow begins by identifying cellular boundaries to segment cells, and then quantifying the marker expressions to annotate the cell types, and then lastly performing spatial statistical analyses. So a critical step in the data process is this step, which is to segment cells from these hyperstacked images. So the proper strategy for performing cellular segmentation utilize algorithms from first the classical image processing algorithms, this literature, or to employ really interactive supervised learning techniques. So with the substantial amount of label data in different modalities in different tissue, with the atlases that are now available, more recent approaches have started to leverage methods that are employing deep learning schemas. So regardless of the method that are being used, the results really highly depend on how you tune the parameters, how you annotate the cells, and lastly, what bank of training data you actually use. So next, I would like to discuss about another challenge of applying segmentation approaches is the identification of common errors. So these common segmentation errors include partially segmenting cells, as we can see in the middle, splitting cells apart in the very right, and then on the very left, which is merging cells in segmentation. So identifying these errors require pathologists to examine each image one by one. So the current canonical approach to evaluate segmentation accuracy is having pathologists look slide by slide to look through the images to assess mass using defined statistical metrics. So while these metrics each assess an aspect of segmentation quality, there are discrepancies in the interpretation of each metric. So in addition, there's a lack of a unified strategy that merges the statistical properties and the biological contexts. So to that end, I present an overview of the new model, the ESQmodel. So this model takes in a protein expression matrix and a prior expression matrix that represents our belief and our expectations of how expressive a certain marker is for a cell type. Later on, we fit the data into our model, and then we calculate an entropy score for each single cell. For well-segmented cells, as we can see, we can expect a lower entropy. And whereas for poorly segmented cells, we can expect a higher entropy. So ESQmodel leverages Markov chain Monte Carlo, MCMC methods to perform Bayesian inference. So our model is a hierarchical model where we define Y as the observed expression across all cells, each with a different marker measurement. We assume the intensity for each marker has its own precision parameter due to antibody properties. We assume that each cell's expression is a contribution of a vector of cell types, where each cell type has its own cell type expression signature. We define that latent vector as rho. We use a metropolis within Gibbs sampler to perform inference, and we derive point estimates of the posterior by simply averaging across the trace. Of course, minus the burn-in. And using the point estimates of rho, we can then compute the entropy per cell. Next, beginning with this slide, I would like to highlight some of the results of the paper. So we first show ESQmodel's utility on 36 IMC images from the Metabrick breast cancer cohort. So each patient's image was manually segmented using elastic 4 times to simulate 1 well, and then 3 erroneous poor segmentations that have merge, partial, and split, as we can see in A. The visualization of how merge and partial and split can be seen in C, where you can see all the probability maps. And then we perform 3 pairwise comparisons between the well and the 3 erroneous conditions in A. We can see an elevated average entropy when errors are introduced into segmentation. And this is also shown in B for an individual case, where you can plot the distributions of entropy in a single IMC sample. So this ultimately, the take-home message is that this demonstrates that entropy can serve as an effective metric to detect segmentation errors. Moving on, next we looked at another interesting case, which is how co-expression of markers affect entropies. So using a segmented data from the original publication from the Metabrick IMC image that was published in Nature Cancer, we explored the relation between spatial distribution of entropies and 4 lineage markers, which is, in this case, CD45, a pan-immune marker, SMA, which is a stromal marker, CK5 and CK7, both luminal and basal markers for breast, and these are immune stromal and epithelial cell types. So we observe a natural partition of the spatial organization by entropy in A, where the top region has higher entropy and the bottom has lower entropy. So we cluster the cells into 4 distinct cell types shown in B and C. And what we can see over here is a correlation between the spatial distribution of entropy and the cluster labeling, where cells that co-express CD45 and CK5, which is an immune marker and an epithelial marker, strongly yield a higher entropy than those expressing mainly a single protein marker. So as the co-expression of these 2 markers, which are lineage markers, are not defined in the prior matrix and are usually not seen, this yields higher entropies. And this exhibits another use case of the ESQmodel, which is to identify potential segmentation errors through well-defined prior matrices, as well as identify cells that deviate from our prior expectations. Okay, it's not moving. Okay. So we also compared the performance of 5 segmentation methods over here, elastic, watershed, and 3 deep learning methods, Stardust, cell pose, and deep cell on IMC images of breast cancer, classical Hodgkin's lymphoma, reactive lymph node, and tonsil. So as a control, as we highlighted here, we did intentional pore segmentation using elastic and Stardust and compared the well to the pore settings. So this was a sanity check where we see that an increase in entropy validates our method's utility. And then for the 4 tissue types, we saw that for breast cancer and reactive lymph node, elastic performed significantly better. For classical Hodgkin's lymphoma, elastic and watershed were better than the deep learning methods. And for tonsil, there was no significantly better candidate. So elastic's better performance is expected because manual segmentation is treated as a gold standard in the field. Watershed can suffer from heterogeneous cell types, having varying cell sizes. And deep learning models depend heavily on their training datasets. So we also plotted heat maps of an exemplar tonsil tissue in B. So we split the heat map into lower 25% entropy and upper 75% entropy. So the top showed a clear pattern of well-defined cells, while the higher entropy exhibited weaker signals. So to showcase the general applicability, we assessed also tonsil images from MIBI, Codex, and cyclic IF. And then we saw a similar pattern between IMC and MIBI, and this is due to the similarity of their mass cytometry-based techniques, while differing from the fluorescence-based Codex and cyclic IF. So Stardust performed the best in the 2 methods, while deep cell performed well in Codex, and watershed was most effective for cyclic IF. So the take-home message over here is that, to conclude, ESQmodel not only can be used as to compare performance, but also serve as a quality control metric to assess whether expression really matches with the prior expectation of protein intensity. All right, so while ESQmodel serves as a new metric for performing quality control, there are limitations to every single model. And I would like to discuss the best use cases for this model. So first being that with more cell types, the uncertainty of cells to be identified as a cell type becomes much larger, and making entropy a less effective metric, as we can see on the left. So the best case is then to limit the cluster count and number of lineage markers. So the fine-grained annotation of cell types can be performed later after images pass quality control. For instance, if you were to label T-cell lineage markers such as CD4 and CD8 for quality control, it's preferable than to put in fine-grained exhaustion markers when we're measuring infiltrating cancer cells, such as LAG3. Second is to choose the correct number of markers is also crucial. So we can see that even if other non-lineage markers are additionally added with an uninformed prior, the average entropy will not be impacted significantly, as shown in the first row, which is the CHL dataset. However, in the tonsil dataset, we can see that the emission of CD68 results in a significantly lower average entropy, which is on the left. And then this can mislead quality interpretation when crucial markers are omitted in the analysis. So to sum it up, we present a novel Bayesian approach, ESQmodel, that can quantitatively evaluate cell segmentation quality using expression data and prior knowledge of expected cell type composition. So ESQmodel takes in and also makes two important contributions to current pipelines in spatial proteomics. The first one being that ESQmodel quantifies single cell segmentation accuracy through biologically informed per cell entropy metric, which can be used to detect the presence of erroneous segmentation. Second is ESQmodel allows for comparative entropy analysis that's instrumental in comparing performances between different segmentation methods on the same dataset. And these contributions can potentially reduce the manual labor of curating segmentation by pathologists. And future work will be to establish a human-in-the-loop software to annotate high entropy cells in segmented images and to integrate this as a plugin for quality control. And lastly, ESQmodel is a general model with the versatility to be applied to other spatial single-cell platforms. So with that, I would like to thank the supervision by the Roth Lab and the Operational Lab, and also data generation by all our MD PhDs, and manual segmentation support and computational inference from the IMAX-D Grand Challenge Consortium. Thank you.
Any questions? Okay, I'll start with one first.
Yep.
Okay, so you mentioned about the challenge as the number of cell types or population increases,
Yep.
it gets somewhat less effective or more
Yep.
challenging. Is there a sweet spot, and or what's the best number of populations?
Yep, so like I quickly talked about, the best use case is when you have defined cell types that are not too large, in a sense that when you're looking only at lineage markers. For example, if you go to T cells, there's T helper 1, T helper 2. You can go even down, you can go even further, Th17, go on. But in the case where you increase a ton of these fine-grained things that is not really the focus of the question of your experiment, these all become like just extra information, extra things that the program has to process, and really just deteriorates the ultimate like goal of how like the entropy function is actually being used. Yeah.
So I guess sort of, is there a number for the too large?
It really depends on like your use case, but as we can see in the graph, probably like 10 markers below 10 markers is fine. But like once you get like your lineage becomes super big, then entropy itself, like the function becomes pretty futile. Yeah.